Welcome to the Deep Dive, the show that takes the complex, dense research materials, distills them down, and delivers you the absolute clearest path to expertise.
And today we are taking on one of the most fundamental and mechanically fascinating tissues in the entire human body.
Muscle. It's the infrastructure of movement.
It really is. I mean, from the massive voluntary efforts that allow you to lift weights, all the way down to the subtle involuntary changes that regulate your blood pressure and digestion.
It all comes down to these specialized cells.
Exactly. Cells built for one thing, contraction. And our mission today is, well, pretty straightforward. We are building a structural blueprint of muscle tissue.
Okay.
We're going to navigate the histology of skeletal, cardiac, and smooth muscle layer by layer, starting right at the molecular foundation.
Okay, let's unpack this then. So, regardless of whether a muscle is moving a bone or, say, constricting a blood vessel, the basic molecular machinery is the same.
That's right. At the core, we're looking at aggregates of specialized elongated cells.
And their whole identity is just changing shape.
So what are the two core molecular players here, the ones responsible for generating that force?
We call them myofilaments, and their interaction is, well, it's the essence of movement. You need to visualize two distinct types here.
Okay.
First up, we have the thin filaments. These are made of a protein called actin.
Actin.
Specifically, you have these little globular actin monomers, G-actin, and they polymerize to form a double helix. That's fibrous actin or F-actin.
I mean, these are small.
About 6 to 8 nanometers in diameter, maybe 1 micrometer long. You can think of them as the tracks.
The tracks. Okay, so what's the engine that runs on those tracks?
That would be the thick filament,
Yeah.
much larger, about 15 nanometers in diameter, 1.5 micrometers long, and they're composed primarily of myosin II molecules.
Hundreds of them, right?
200 to 300 of them, all aggregated together. The best way to picture a myosin II molecule is it has this long coiled tail,
Yeah.
which bundles up with other tails, and then it has two globular heads that project outward. And those are the bits that do the work.
Those are the bits. It's the action of those myosin heads binding to and pulling on the actin tracks that generates mechanical force.
And we should probably establish the specialized vocabulary right up front. The cytoplasm in a muscle cell is always called the sarcoplasm.
Right. And that name reminds you that inside these cells, the contractile machinery, these myofilaments, they take up almost all the space. There are highly, highly differentiated cells.
The specialization is really the key, isn't it?
It is. I mean, actin and myosin exist in tiny amounts in almost all your cells for basic stuff like cell division. But in muscle tissue, they are packed in and organized for one single purpose.
Contraction.
Contraction.
Now, we classify muscle based on how it looks under a microscope. And this gives us two major families, right? Striated and smooth.
Exactly. And the difference is all about organization. Striated muscle has those, dramatic, repeating cross-striations.
That's right.
The stripes, yeah. And they're there because the myofilaments are laid out with just military-grade precision. It creates this predictable repeating pattern called the sarcomere.
And even within striated muscle, we subclassify it based on location and how it's controlled.
We do. There are three subtypes. First is skeletal muscle. This is what you generally think of when you hear the word muscle.
Attached to the bones.
Right, attached to the skeleton,
Right.
responsible for posture and voluntary movement. It also includes really specialized, precise muscles, like the extraocular muscles that move your eyes.
Okay, what's the second type?
That's visceral striated muscle. Morphologically, it looks identical to skeletal muscle, but you find it in soft tissues. Think tongue, pharynx, the diaphragm, upper esophagus.
So crucial for things like speaking and swallowing.
Exactly. And the third type is cardiac muscle.
The heart.
Confined pretty much exclusively to the heart wall and the base of the big veins that feed into the heart.
Which leaves us with the other major family, smooth muscle.
Smooth muscle is the non-striated one. It lacks those cross-striations because its myofilaments are just less rigidly organized. There are no sarcomeres.
And this is the regulatory muscle.
That's a perfect way to describe it. You find it restricted to the viscera, the vascular system controlling blood flow, the tiny erector pili muscles that give you boost bumps, and the intrinsic eye muscles that control your pupil size.
All right, let's start our deep structural dive with skeletal muscle.
Okay.
The fundamental unit here isn't just a cell, it's the muscle fiber, which is this giant thing Austin described as a multinucleated syncytium.
And that term, syncytium, is so important. During embryonic development, these individual precursor cells called myoblasts, They fuse together.
End to end?
End to end, and this fusion creates one massive continuous cell, the muscle fiber, that inherits all the nuclei from the myoblasts that formed it.
Which makes sense. You'd need all those nuclei to pump out the huge amounts of protein for the contractile machinery.
Precisely. And the size variability is it's astonishing. These fibers are typically 10 to 100 micrometers in diameter.
But the length can be huge.
It can, from millimeters, like in the tiny stapedius muscle in your ear, up to nearly a meter in the sartorius muscle of the thigh.
And their internal structure is really distinctive. A key visual giveaway for skeletal muscle is the location of all those nuclei.
It is. They're not in the middle. They're pushed right out to the periphery, lying immediately beneath the plasma membrane.
Which we call the sarcolemma.
The sarcolemma, right.
Yeah.
Which is the membrane plus its external and reticular laminae. Seeing those peripheral nuclei is one of the quickest ways you can distinguish skeletal from cardiac muscle on a tissue slide.
Okay, so a muscle fiber, no matter how powerful it is pretty useless if its force can't be transmitted to the bone.
Right.
This requires a pretty spectacular system of structural support, the connective tissue organization. And this is what we're seeing in figure 11.2.
Yes, and it's essential for force transduction. This connective tissue acts like a series of nested sheaths. They hold the fibers together and integrate them into the tendon.
So you work from the individual fiber outwards. Let's start with that innermost layer, the most delicate wrapping.
That's the endomysium. It's made of fine reticular fibers that surround and support each single muscle fiber.
In figure 11.2a, that freeze fracture image, it shows this perfectly. It looks like a fine basket weave structure around each cell.
It's a beautiful image. And this layer is where you find the capillary beds, the tiny blood vessels, and the very finest branches of the motor neurons.
Okay, so moving up a layer, how are these fibers bundled together?
They're grouped into units called fascicles. And the layer that does the bundling is the perimysium.
And this is a thicker layer.
A much thicker layer of connective tissue. The fascicle is the functional unit. It's a group of fibers that often performs a specific action. And the perimysium is where you find the larger blood vessels and nerves that then branch out to feed the individual fibers.
And then the entire muscle gets its own wrapper.
The epimysium. This is a dense connective tissue sheath around the whole collection of fascicles.
So this is what anatomists call the deep investing fascia.
That's the one. It's the entry point for the major vascular and nerve supplies, and it merges seamlessly with the tendon at the end of the muscle.
Now, speaking of function, not all skeletal muscle fibers are the same. There's a whole classification system based on their performance profile.
Right. It's based on how fast it contracts, the speed of its myosin ATPase enzyme, and whether it relies more on oxidative phosphorylation versus glycolysis.
It's really where you see the biological design priorities at work.
Absolutely. The performance depends entirely on its metabolic machinery. Fibers that are high in oxygen carrying capacity look red, while the ones that rely on quick sugar bursts look white.
And the protein responsible for that internal oxygen storage is myoglobin.
Myoglobin, yep. It's a small protein with an iron atom, F2 plus AT, that binds and stores oxygen right there in the muscle fiber, ready for the mitochondria to use.
And the concentration of myoglobin is what dictates that color.
Right. High myoglobin means a dark red fiber.
This color contrast links directly to a pretty severe clinical connection, rhabdomyolysis.
Ah, yes. When a patient has a severe crush injury, a huge amount of skeletal muscle gets destroyed.
And all that stored myoglobin gets released into the bloodstream.
Exactly. The kidneys try to filter it out, but myoglobin is actually toxic to the renal tubules, and a massive dose can quickly lead to acute renal failure.
A really serious consequence of cellular level damage.
It is.
So when we're classifying these three fiber types, we often use histochemical reactions because they show the metabolic differences more clearly than a simple stain.
Right, like stains for NADHTR or succinic dehydrogenase activity.
And figure 11.3 shows that visual contrast perfectly, the deeply stained oxidative fibers versus the lighter glycolytic ones. Let's break down the three types.
Okay, first, type I. These are the slow oxidative fibers.
The endurance athletes.
The endurance athletes, exactly. They're small, they look red, and they are just packed with mitochondria, myoglobin, and cytochrome complexes. They rely almost totally on efficient oxidative phosphorylation.
So they're slow twitch?
Slowest myosin ATPase. So, yes, slow twitch. But, and this is critical, they are incredibly fatigue resistant. They're perfectly adapted for sustained work, like maintaining your posture. Think about your erector spinae muscles.
Or long distance running.
Or long distance running. Their design priority is efficiency and longevity.
Got it. What's the intermediate type?
That's Type IIa, the fast oxidative glycolytic fibers.
So a hybrid approach.
It's a hybrid. They're medium-sized, intermediate in color. They have lots of mitochondria and myoglobin, so they have decent fatigue resistance.
But they can also do glycolysis.
Right. They have large glycogen stores and can quickly switch to anaerobic glycolysis for a burst of power. They're fast twitch, generate high peak tension, and are ideal for things like swimming or middle distance running.
And finally, the pure sprinters.
Type IIb, the fast glycolytic fibers.
These are the big ones.
They're the largest fibers. They look light pink or white because they have very little mitochondria or myoglobin. They are all about brute force and immediate speed.
So fast as myosin ATPase.
The fastest. They rely almost entirely on anaerobic enzymes and their glycogen stores. So their fast twitch generate the highest peak tension, but they are also very fatigue prone.
Because of that metabolite buildup.
Exactly. They're adapted for rapid, fine, precise movements, like your extraocular muscles, or maximal power outputs like weightlifting or short sprints.
It's a perfect biological specialization. You're trading metabolic efficiency for speed and power.
It is. And you really feel this difference when your Type IIb fibers kick in for a surprise sprint and they fatigue so quickly.
Okay, now we drill down inside the muscle fiber to the actual components that create those striations we talked about.
Myofibrils.
Myofibrils.
These are structural subunits that run the length of the fiber, and they're essentially just bundles of myofilaments.
And they're surrounded by the sarcoplasmic reticulum.
Right, the SCR, and interspersed with mitochondria for energy.
And this precise longitudinal array of myofibrils is what creates the visually defining cross-striation.
That's it, the alternating light and dark bands.
The dark bands are the A bands.
A for anisotropic, or birefringent and polarized light. And the light bands are the I bands.
I for isotropic.
Correct. Now, when we zoom in with an electron microscope, like in figures 11.4 and 11.5, we see even finer details that define this whole architecture.
So what are those interior landmarks?
The I band, the light one, is bisected by a dense structure called the Z line, or Z disk.
Okay.
The A band, the dark one, is bisected by a lighter region called the H band. H for hell, which is German for light.
And then right through the middle of that H band
is a narrow, dense line called the M line, M for midi, German for middle.
And the segment between two of those adjacent Z lines is the functional contractile unit.
The sarcomere. That is the definition.
The sarcomere.
It's the smallest functional unit of the whole apparatus, measuring about 2 to 3 micrometers when it's relaxed. And the fact that all the sarcomeres and all the adjacent myofibrils are perfectly aligned.
That's what gives the whole fiber its striped pattern.
That's what does it.
Okay, let's talk about how the filaments are actually positioned to create this banding pattern. Figure 11.6 has some great diagrams for this.
So the thick filaments, the myosin, are confined entirely to that central A band. Their length is constant, about 1.6 micrometers.
They don't change length.
They do not. The thin filaments, the actin, they attach to the Z line and extend inward, overlapping with the thick filaments in the outer parts of the A band.
So to be clear, the I band contains only thin filaments.
Correct.
And that central H band contains only thick filaments.
You got it. And this arrangement is the basis for the sliding filament model of contraction.
Right.
When the muscle contracts, the myosin heads pull the actin filaments deeper inward. So the I band and the H band shorten dramatically.
But the width of the A band, the length of the myosin filaments, stays the same.
It remains unchanged. It's a beautiful geometric efficiency. You can see it in cross-section, where in that overlap zone, each thick filament is surrounded by a perfect hexagonal array of 6 thin filaments.
Now we need to look even closer at the thin filament components from figure 11.7, because actin is really only half the story here.
It is. The regulatory elements are essential.
Okay.
So beyond the F-actin double helix, you have proteins that stabilize it and proteins that act as switches. The F-actin itself is polarized.
Meaning it has a plus and a minus end.
Exactly. The plus or barbed end is anchored firmly to the Z line by a protein called alpha-actinin. The minus or pointed end is capped and protected by another protein, tropomodulin.
And what about the regulatory components, the ones that decide if a contraction can even happen?
That's the tropomyosin-troponin complex.
Yeah.
Tropomyosin is a double helix that kind of snakes along the groove of the F-actin. Now, in a relaxed muscle, tropomyosin physically masks the binding sites that myosin needs to grab onto.
So it's blocking the connection. And the troponin complex is the trigger itself.
Is the trigger. The complex is attached to tropomyosin, and it has 3 vital subunits.
Where are they?
First is TNT, troponin T. That just anchors the complex to tropomyosin.
Simple enough.
Then you have TNI, troponin I. The I is for inhibitor. It binds to actin to prevent the interaction with myosin. This is the molecular brake.
And the third one must be the sensor.
That's TNC, troponin C. The C is for calcium. This is the central sensor. When calcium is released into the sarcoplasm, it binds to TNC.
Which causes a shape change.
A big conformational shift that yanks tropomyosin away, unmasking the myosin binding sites.
So calcium pulls the lever, tropomyosin moves the mask, and TNI is overridden. It's a beautifully coordinated dance.
It is, and we should really reiterate the clinical significance of these proteins.
TNI and TNT.
Right. They have specific cardiac isoforms. And because these are highly specific to heart muscle, their presence in the bloodstream is the gold standard diagnostic marker for a heart attack.
Myocardial infarction.
Right. It tells you unequivocally that cardiac muscle cells have been damaged and have leaked their contents.
And there are also structural helper proteins in the thin filament, like nebulin.
Nebulin. Nebulin is essentially the molecular ruler of the thin filament.
A ruler.
Yeah, it's a gigantic, non-elastic protein that spans the entire length of the thin filament. It confirms its length and anchors it to the Z line right alongside alpha-actinin. That stabilization is vital for maintaining that precise sarcomere distance.
Okay, let's turn to the thick filament components, the myosin, the second motor itself, which we see in figures 11.8 and 11.9.
The myosin, the second motor, is a dimer. It has 2 heavy chains and 4 light chains. And we need to distinguish its functional parts. You've got the 2 globular S1 heads, that's the business end. They house the ATP binding site, which gives it ATPase activity, and the actin binding site.
And they're connected to the tail by a lever arm.
Right, the S2 lever arms connecting to the long tail. And the light chains, the ELC and RLC, they're crucial for stabilizing that lever arm structure.
And the aggregation of these individual myosin molecules is what creates that bipolar thick filament.
Exactly, they aggregate tail to tail, which is why the myosin heads project outward helically in both directions, leaving that centralized.
Bare zone.
The bare zone, free of heads. And that bare zone is anchored by the proteins of the M line.
This brings us to the just. Astonishing team of accessory proteins in figure 11.10 that are required just to keep this whole structure from flying apart under tension.
You can think of them as the biological scaffolding and suspension system. It's incredible.
Okay, let's start with Titin.
Titin is the molecular spring, or bungee cord. It's an enormous elastic protein. It spans half the sarcomere from the Z line all the way to the M line.
So what does it do?
It prevents the muscle from being overstretched, and it helps recenter the thick filament passively during relaxation. It's a spring.
Then you have alpha-actinin and desmin.
Alpha-actinin is the main anchor at the Z line, and desmin is an intermediate filament that forms a lattice around the Z lines, physically linking adjacent myofibrils to each other and to the sarcolemma.
So it ensures synchronous contraction across the whole cell.
Precisely. Then you have the M-line proteins, like myosin, whose sole purpose is to hold the thick filament securely in a register at the center.
And then there's dystrophin.
Dystrophin is the absolute crucial structural link to the outside world.
Okay, let's focus on dystrophin. It links the internal actin cytoskeleton to the extracellular matrix via laminin. Why is this anchor bridge so vital?
It's the critical transmission point. It takes the internal contractile force and anchors it to the external environment. Dystrophin itself is this rod-shaped protein right under the sarcolemma.
And it forms a whole complex.
The dystrophin glycoprotein complex. This entire assembly ensures that when the actin filaments contract, the force is transmitted laterally to the external lamina, stabilizing the cell membrane during the extreme stress of contraction.
And when this anchor bridge fails, we see the devastating consequences of the muscular dystrophies.
This is a classic example of structure-dictating pathology. In Duchenne muscular dystrophy, DMD, mutations cause the complete absence of functional dystrophin.
But without that anchor,
the cell membrane is incredibly fragile. It gets damaged during every single contraction cycle. This leads to repeated injury, degeneration, and eventually the muscle is replaced by fat and fibrous tissue.
which is why the progression is so rapid.
It is. Unfortunately, most affected boys lose the ability to walk by early adolescence. Becker muscular dystrophy is a similar but slower form.
And this really highlights why so much therapeutic research is focused on gene therapy, just trying to replace that one missing structural element.
Exactly. If you can't anchor the contraction, the muscle literally self-destructs.
Okay, let's turn to the actual physical motion. The actomyosin cross-bridge cycle, shown in figure 11.11.
Mhm.
This is the precise sequence of events that uses ATP to power the sliding of the filaments.
And we need to be clear, this cycle only starts when calcium has bound TNC and cleared tropomyosin out of the way.
Right, so what are the 5 stages?
Stage 1 is attachment. The myosin head is tightly bound to actin. We call this the rigor configuration. ATP is absent.
And this state is responsible for rigor mortis after death.
It is. When the ATP supply fails, all the myosin heads get stuck in this attached state.
Okay, what's stage 2?
Release. A new ATP molecule binds to the myosin head. The mere binding of ATP causes a shape change that reduces myosin's affinity for actin.
And it lets go.
It lets go. Stage 3 is bending, or the recovery stroke. The ATP is hydrolyzed into ADP and pi, which stay bound. The energy from that hydrolysis causes the myosin lever arm to rotate into its high-energy cocked position.
It's ready to fire.
It's ready to fire. Stage 4 is force generation, the power stroke. The cocked myosin head weakly binds to a new site on the actin. Then the release of that inorganic phosphate, pi, rapidly increases the binding affinity.
And the head snaps back.
It snaps back, pulling the thin filament along with it. That is the power stroke. Then the ADP is lost.
And the final stage.
Reattachment. The myosin head is once again tightly bound to the new actin site, back in the rigor configuration, waiting for the next ATP molecule to restart the whole process.
And it's the rapid, coordinated repetition of these 5 steps that allows the sarcomere to shorten.
Billions of them, all at lightning speed.
Okay, so that whole cross-bridge cycle is completely dependent on the precise and instantaneous release and removal of calcium.
Absolutely. And that regulation is handled by 2 specialized membrane systems, the sarcoplasmic reticulum, or SR, and the transverse tubular system, the T system.
So the SR is the internal calcium warehouse. How is it structured within the fiber?
The SR is this highly organized internal network that wraps tightly around the myofibrils, and as the junction between the A band and the I band.
The AI junction?
Right. At the AI junction, the SR forms these enlarged ring-like reservoirs called terminal cisternae. These are the actual storage tanks for the calcium.
And the T system is what transmits the signal from the cell surface deep into the core of the fiber.
Exactly. T-tubules are direct invaginations of the sarcolemma. They penetrate the fiber transversely, and they run precisely between 2 adjacent terminal cisternae at that AI junction.
And that highly specialized structure. 1 T-tubule sandwiched between 2 terminal cisternae.
That's the triad. You can see it in figure 11.12. And in skeletal muscle, you have 2 triads per sarcomere.
So what's the mechanism that links the electrical signal on the T-tubule to the calcium release from the SR? It's not just proximity, right?
It's a physical linkage. It's mechanical. The T-tubule membrane contains these dihydropyridine-sensitive receptors, or DHSRs. They're voltage sensors.
Okay.
When the electrical impulse arrives, the DHSRs physically change their shape. And this conformational change directly activates the mechanically gated ryanodine receptors, the RR1s, which are located in the adjacent SR membrane.
So one physically pushes the other open.
It's a mechanical handoff of the signal, and it causes an explosive rapid release of all that stored calcium.
And to store the enormous concentrations of calcium needed for this, the SR needs help preventing a massive osmotic gradient.
That is the job of calsequestrin. This is a highly acidic protein inside the SR lumen, and it can bind up to 50 calcium ions per molecule.
It's a buffer.
A massive buffer. It keeps the concentration of free calcium in the lumen low, which is crucial for reducing the energy demand on the calcium pumps that are constantly working to bring calcium back into the SR for relaxation.
Okay, let's run through the full 10 steps of the depolarization cascade. This is excitation, contraction, coupling in action.
Let's do it. 1. Nerve impulse arrives at the neuromuscular junction, the NMJ.
2. Acetylcholine, HE8, is released and binds to its receptors, causing a local depolarization.
3. Voltage-gated sodium channels open, and that depolarization spreads along the sarcolemma.
4. The impulse travels deep into the fiber via the T-tubules.
5. The DHSRs in the T-tubule membrane sense the voltage and change their conformation.
6. This change physically activates the RYR1 calcium release channels in the SR.
7. Calcium is released rapidly into the sarcoplasm.
Wait, calcium binds to TNC.
9. The actomyosin cross-bridge cycle is initiated, and the muscle contracts.
And for relaxation, step 10. Calcium is actively pumped back into the SR by ATPase pumps, where it's captured again by calsequestrin.
And that sequence explains the sheer speed of voluntary muscle response.
Right. Now, the initiation point of that whole sequence is motor innervation. specifically the neuromuscular junction, which is detailed in figures 11.13 and 11.4.
The NMJ, or motor end plate, is that precise contact area. The axon terminal loses its myelin sheath,
Okay.
and the nerve ending sits in a shallow depression on the muscle fiber. The terminal is just loaded with vesicles full of acetylcholine ready to go.
And the postsynaptic structure on the muscle side is also highly specialized.
Absolutely. The muscle fiber membrane features these deep junctional folds, and this massive folding drastically increases the surface area.
And the receptors are all concentrated there.
The nicotinic 8 receptors are heavily concentrated right at the top of these folds. This ensures that even a small release of H-ray causes a massive localized sodium influx and a very efficient depolarization.
And then the signal has to be shut off quickly.
Right. Immediately after binding, the enzyme acetylcholinesterase, H3, is right there in the synaptic cleft to break down the H3 rapidly. This ensures the signal is brief and precise.
The delicate nature of this process is really highlighted by the clinical condition myasthenia gravis.
This is a classic autoimmune disease. The body mistakenly produces antibodies that bind to and block the functional NATO receptors.
So the signal can't get through.
It's drastically reduced. Because the depolarization signal relies entirely on these receptors, blocking them just cripples the muscle's ability to respond. The initial symptoms often show up in the extraocular muscles, leading to drooping eyelids or double vision.
And the treatment involves trying to support the neurotransmitter that's already there.
Exactly. Since the number of receptors is reduced, the strategy is to prolong the life of the AC that is released. So you administer ATE inhibitors.
To slow down the breakdown of acetylcholine.
Right. It gives it a better chance to find one of the few remaining functional receptors.
We should also briefly mention how potent certain pharmacologic blockades are at this site.
Oh, the NMJ is highly targetable. Botulinum toxin blocks the release of HF vesicles entirely,
Causing paralysis.
right. And poisons like curare bind to the receptor without opening the channel, also leading to immediate paralysis, including respiratory failure.
This precision of control is defined by the motor unit.
A motor unit is one single motor neuron and all the muscle fibers it innervates, and the ratio is functionally adapted.
Right. So for fine control, like in the eye, you have a very low ratio.
One neuron controls just a handful of fibers, but for big, powerful postural muscles, like the gastrocnemius in your calf, the
Right.
ratio is very high. One neuron controls thousands of fibers.
And that innervation is trophic, right? It keeps the muscle healthy.
It does. If you sever the nerve, the muscle tissue atrophies severely.
Now, skeletal muscle doesn't just receive motor input. It also provides sensory feedback.
Proprioception. The brain needs to know what the muscles are doing.
And this involves two primary types of specialized receptors.
The muscle spindles and the Golgi tendon organs.
Let's start with the muscle spindle from figure 11.1c. This is the muscle's internal odometer.
That's a great analogy. The muscle spindle is encapsulated and located right within the muscle belly. It contains these modified muscle fibers wrapped by sensory afferents.
And what are they sensing?
They monitor the muscle's length and, importantly, the velocity of stretch, so how long it is and how fast it's changing length.
So they provide that stretch reflex information.
They do.
Yeah.
And they also receive gamma efferent fibers, which regulate the sensitivity of the receptor itself, keeping it functional no matter how contracted the muscle is.
In the Golgi tendon organs.
These are located in the tendons, not the muscle belly, and they monitor muscle tension or the force of contraction.
A safety mechanism.
It's a protective feedback mechanism. They send signals to prevent the muscle from generating so much force that it could damage the tendon or the bone it's attached to.
Finally, let's talk about development, repair, and renewal. Skeletal muscle has a limited but really important ability to heal itself.
And that's thanks to satellite cells.
Right. So myoblasts are the building blocks, driven by the MyoD transcription factor.
And myostatin is the brake, the negative regulator of growth.
But it's the satellite cells, which we see in figure 11.17, that are the muscle stem cell pool that allow for repair in adults.
These are quiescent stem cells marked by PAX7 expression, and they're just nestled between the sarcolemma and the external lamina.
And when an injury occurs,
They activate. They start expressing MyoD, they proliferate into new myoblasts, and those can fuse to form new fibers. But, and this is a big but, this process is only successful if the external lamina remains intact to provide a scaffold.
if it's too damaged, you just get scar tissue.
Exactly. Fibroblasts rush in, and the site is repaired with non-functional scar tissue.
Okay, that brings us to the functional considerations of metabolism in folder 11.1. The efficiency and speed of contraction we've been talking about are dependent on a constant supply of ATP.
A constant managed supply. ATP and phosphocreatine are the immediate energy sources, and they're derived from the metabolism of fatty acids and glucose.
And for rapid, intense contraction, it's all about glucose.
Right, from the circulation or from stored glycogen.
And when those type I fibers are firing hard, they're relying on anaerobic glycolysis, which leads to a high metabolite buildup. This relates back to cramps or ischemic pain.
Right, and the old school thought was just blame lactic acid.
But it's more complicated than that.
It is. Current research gives us a more sophisticated view. While lactic acid does cause a pH drop, the pain signal appears to be a combined effect. ATP released from the ischemic muscle cells works synergistically with the lactic acid.
That's so.
It sensitizes a specific channel on the sensory neurons, the acid-sensing ion channel 3 or AC3. It's the coaction of these molecules that sends that intense pain message to the brain, not just the lactic acid alone.
A brilliant example of how structural biology dictates sensation. And conversely, when the muscle is resting or recovering, it switches back to the most efficient energy source.
Oxidative phosphorylation. This process primarily uses beta-oxidation of fatty acids inside the mitochondria, and this high-efficiency long-term process relies on a steady supply of oxygen from hemoglobin and, crucially, from the oxygen stored locally in myoglobin.
All right, let's transition now to the heart muscle, cardiac muscle. It shares the striated appearance and the sarcomere structure with skeletal muscle, but its functional organization is entirely different. It's all about tireless rhythm, not voluntary speed.
The cardiac myocytes are structurally very distinct, as you can see in figure 11.19. They are individual cylindrical cells arranged end to end, and they frequently branch.
And they're much smaller than skeletal muscle fibers.
Much smaller. About 15 micrometers in diameter and maybe 80 micrometers long.
The nuclei are a key differentiator as well.
They are. Cardiac cells typically have a single, centrally located nucleus. You might find a binucleated cell, but that central positioning is instantly recognizable and distinguishes them from the peripheral nuclei of skeletal muscle.
And if we look closely at the space right around the nucleus, the juxtanuclear region in figure 11.20, what do we find?
You find the cell's metabolic power station. The myofibrils literally part ways to go around the nucleus, leaving this biconical region just crammed with organelles.
Especially mitochondria.
Many, many large, densely packed mitochondria, along with Golgi and glycogen stores. The sheer concentration of mitochondria reflects the heart's relentless, non-stop need for ATP.
And the atria also contain specialized hormonal granules.
Right. Atrial myocytes contain atrial natriuretic factor, ANF, and brain natriuretic factor, BNF.
And these are diuretics.
They are. They promote sodium excretion and regulate blood volume and pressure by inhibiting renin and aldosterone. BNF levels are particularly important clinically as they rise significantly in congestive heart failure.
The defining histological feature of cardiac muscle has to be the intercalated disc, the ID. This is where the cells join up structurally and electrically.
The intercalated discs, shown in figure 11.21, are those densely staining cross bands. They often appear in a step-like configuration, and we need to analyze the three main components that make them up.
Okay, so what's handling the mechanical welding? What holds them together?
That's primarily the fascia adherens and the maculae adherentes, or desmosomes. The fascia adherens anchors the thin filaments of the very last sarcomere directly to the plasma membrane. And the desmosomes act as spot welds, tightly binding the cells together to prevent them from separating under the immense repetitive stress of the cardiac cycle.
And what's the key element for the electrical coordination?
Gap junctions. Communicating junctions. These are concentrated mainly in the lateral component of the disc, which is a position that's shielded from the maximum contractile stress.
And these provide ionic continuity.
They do. They allow the rapid passage of ions and electrical signals from one cell to the next, turning all these individual cells into a coordinated functional syncytium.
Okay, let's look at the regulation of contraction in cardiac muscle. The molecular machinery is striated, but the architecture of the calcium regulation system is unique.
It is. The SR is less organized than in skeletal muscle. And crucially, the T-tubules penetrate the cell at the Z-line, not the AI junction.
So there's only one T-tubule per sarcomere.
One per sarcomere. And this single T-tubule pairs with only one small SR terminal cisterna, forming what we call a dyad.
A dyad, not a triad. Why does the heart need this specific structure?
It relates to the activation mechanism, which is called calcium-triggered calcium release. You see it in figure 11.22.
So, unlike skeletal muscle, The heart can't contract using only its internal calcium stores.
It cannot. It needs a trigger from the outside.
So it's a chemical key and lock mechanism, not a physical handoff.
Exactly. When the depolarization wave arrives, the DHSRs in the T-tubule membrane act as functional calcium channels. They open, and a small but vital influx of calcium comes in from the extracellular space.
And that incoming calcium is the trigger.
That is the ligand that binds to and activates the RIR2 receptors in the SR membrane, causing the massive internal calcium release that starts the cross-bridge cycle. This reliance on external calcium gives the heart a key mechanism for modulation.
And just as we saw with RIR1 in skeletal muscle, mutations in RIR2 are clinically very dangerous.
They are. Mutations in RIR2 are associated with a severe inherited condition, a stress-induced polymorphic ventricular tachycardia. A defect in this calcium release channel can cause uncontrolled, dangerously high heart rates, particularly under stress, which can lead directly to sudden cardiac death.
The rhythmic nature of the heart is also dependent on a specialized conducting system.
The heart's beat is intrinsic, it's spontaneous. It's initiated and coordinated by specialized cells called Purkinje fibers. You can see them in plate 11.5.
And these are different from normal myocytes.
They're larger. Their myofibrils are often sparse and pushed to the periphery, leaving a clear cytoplasm because of abundant glycogen stores. Now, the autonomic nervous system can modulate the rate.
Speed it up or slow it down.
But it never initiates the contraction. That's all intrinsic.
Finally, injury and repair. The prognosis for damaged heart muscle is notoriously poor.
The primary mechanism of injury is myocardial infarction, a heart attack caused by ischemia. When cardiac myocytes die, the tissue undergoes necrosis.
And it gets replaced by scar tissue.
By fibrous connective tissue, a scar. And that scar has no contractile function, resulting in a permanent loss of cardiac output.
Though research is slowly shifting the historical view that regeneration is entirely impossible.
It is. I mean, while major repair results in scarring, there is evidence of very low rates of myocyte mitosis. Carbon-14 dating confirms a slow turnover of about 1% per year in young adults. The problem is that attempted repair often results in binucleated or polyploid cells, cells that get bigger but fail to complete division. So the functional muscle mass is rarely replaced after a significant injury.
All right, let's shift to our final type, smooth muscle, which gets rid of the sarcomere entirely and opts for a design philosophy centered on efficiency and sustained tension, not speed.
Smooth muscle occurs in sheaths or bundles of elongated fusiform cells with tapered ends. You see them in figure 11.23. Their size is highly variable.
And they're designed for slow, prolonged contraction.
Maintaining what we call tone, and their coordination relies heavily on structural communication.
The gap junction.
Abundant gap junctions, or nexus. These provide direct pathways for ions and signaling molecules, ensuring that the entire sheet of muscle contracts in a synchronized wave. It's essential for things like peristalsis.
Histologically, the nucleus provides a really strong clue as to the muscle's state.
It does. The nucleus is central and elongated when the cell is relaxed, but when the smooth muscle contracts, the whole cell shortens and twists.
And the nucleus gets crinkled.
It gets forced into a characteristic wrinkled corkscrew appearance. This is an invaluable visual identifier, especially when you're trying to tell smooth muscle cells apart from fibroblasts.
Their contractile apparatus is fundamentally different from striated muscle, especially the regulatory proteins.
The thin filaments still have actin and tropomyosin, but, and this is a huge point, smooth muscle lacks the troponin complex entirely.
No troponin.
No troponin. Instead, regulatory control comes from two smooth muscle-specific proteins called Blasemin and calconin, which block the myosin binding site in the relaxed state.
And the thick filaments also have a unique architecture.
They're made of smooth muscle myosin, SMM, which aggregates into side-polar, non-helical, thick filaments. You see this in figure 11.26.
So instead of that central bare zone like a skeletal muscle,
there is no central bare zone. The SMM molecules are staggered so the heads point in one direction along one half and the opposite direction along the other.
That sounds like a much more versatile design.
It is. The absence of the bare zone maximizes the length over which actin and myosin can interact, which is necessary for the smooth muscle cell to shorten so dramatically and generate prolonged force.
So where do these filaments anchor if there are no Z-lines?
They anchor to structures called dense bodies, which are in figure 11.25. These are alpha-actinin-containing structures scattered throughout the sarcoplasm and attached to the sarcolemma. Functionally, they are the intracellular analogs of Z-lines.
Okay, let's discuss the initiation of contraction from figure 11.28. Since there's no TNC, the regulatory switch has to be on the thick filament.
Correct. All smooth muscle stimuli, whether they're mechanical, like a passive stretch, electrical from a nerve, or hormonal and chemical, they all ultimately lead to a rise in cytosolic calcium.
And that calcium activates the cascade.
It does. First, calcium binds to the calmodulin protein.
Okay.
Second, the calcium calmodulin complex activates an enzyme, Myosin Light-Chain Kinase, or MLCK.
And what does MLCK do?
Third, MLCK phosphorylates the myosin regulatory light chains. That phosphorylation is the switch. It changes the myosin from an inactive folded state to the active side polar configuration, and that initiates the cross-bridge cycle.
The activation is much slower than in skeletal muscle, which matches its functional requirement for slow, prolonged contraction.
Right. The SMM hydrolyzes ATP at only 10% the rate of skeletal muscle, which is why it's slow but incredibly ATP efficient. And this leads us to the most vital functional characteristic of smooth muscle, the latch state.
The latch state. This allows organs like blood vessels to maintain background tension or tone almost indefinitely without consuming excessive energy. How does this mechanism work?
It's a mechanism to maintain force while essentially hitting the energy brake. So after the initial phosphorylation and contraction, the attached myosin head gets dephosphorylated by another enzyme.
Okay.
This dephosphorylation prevents the myosin head from detaching rapidly. It stays tightly bound to actin in the rigor state, but because it lacks the phosphate, its ATPase activity drops dramatically.
So the tension is sustained, but the ATP consumption just plummets.
It plummets. It is an ingenious evolutionary solution for sustained mechanical work.
And because smooth muscle controls organs that need constant modulation, the innervation is different, relying on the ANS and hormones.
Right. ANS neurons release neurotransmitters from these enlargements in their axons called boutons en passant. And because there's no tight NMJ, the transmitter has to diffuse 10 to 20 micrometers to reach the receptors.
Explaining the slower response time.
Exactly. And once the signal reaches one cell, it propagates rapidly through the gap junctions to the whole sheet. And they're also highly sensitive to hormones like oxytocin and epinephrine.
We also mentioned that smooth muscle has a secretory function.
It does. These cells are highly metabolically active. They synthesize the surrounding connective tissue matrix collagen, elastin, proteoglycans. They have a well-developed RER and Golgi. In blood vessels, they're the dominant source of collagen and elastin.
And finally, their capacity for renewal, repair, and differentiation is far superior to striated muscle.
Smooth muscle cells retain the capacity for mitosis and proliferation throughout life. We see this dramatically in the uterus during pregnancy and constantly during vascular repair. New cells can even differentiate from vascular parasites or convert from fibroblasts.
So when we do a final functional comparison of these three muscle types, the structural differences really do tell the functional story.
They do. And cardiac muscle is, in a way, an evolutionary compromise. It uses the speed and organization of striations from skeletal muscle, but it adopts the propagation and efficiency strategies of smooth muscle.
Okay, let's quickly review the key differences that allow for instant identification and define their function.
For skeletal muscle, you're looking for peripheral, multinucleated cells, triads at the AI junction, and TNC regulation acting on actin. It's fast and voluntary.
For cardiac muscle,
Central, single, or binucleated cells, dyads at the Z-line, still TNC regulation acting on actin, but it's spontaneous, rhythmic, and requires external calcium.
and for smooth muscle,
Central, single, corkscrew nucleus, no T-tubules, uses caveolae, and calmodulin MLCK regulation acting on myosin. It's involuntary, slow, and sustained.
the ultimate distinction, as we've noted, really lies in that regulatory switch.
It does. Skeletal and cardiac muscle regulate contraction via the actin filament using the troponin complex. Smooth muscle regulates contraction via the myosin filament using the calmodulin MSCK system.
And that decision where to put the regulatory switch defines whether the muscle is built for rapid disposable power or for energy-efficient sustained tone.
That's the bottom line.
We've taken a comprehensive deep dive into the three engines of the body, examining the molecular scaffolding required for skeletal muscle's explosive power, the unique electrical demands of cardiac muscle's tireless force, and the profound efficiency that drives smooth muscle's slow, sustained tension.
The structural investment is just enormous, but the payoff is all of human mobility and physiological regulation. It really is a difference between a high-performance engine at max output And a slow, sturdy gear that never breaks down.
And as a final thought for you to appreciate the engineering brilliance here, reflect on that latch state one more time. That ability in smooth muscle to maintain tension by decoupling ATP consumption from force generation stands as one of the most remarkable evolutionary adaptations in the body.
It really does. It proves that biological systems don't always need rapid myosin cycling to solve a problem. Sometimes the best solution is simply a molecular rigor mechanism built for pure efficiency.
A profound study in biological engineering indeed. Thank you for joining us for this deep dive. We hope you feel thoroughly equipped with the structural blueprint of muscle tissue.
Until next time, keep digging deep for knowledge.